would you introduce yourself, give us a little bit of your background, and what caused you to either start your company or to get involved with the company?
Yeah. So my name is Erhard Fernholz, and I'm chemist by training. I've synthesized macrolides completely. I made a postdoc at Scripps Clinic in La Jolla near San Diego in immunology. And then I worked over 20 years with Roche Diagnostics, developed over 30 products, and have had we're in different positions there. Most important, I worked already there in the cancer diagnostics field. And I have a high interest in cancer diagnostics because simply I have a number of cancer cases in my family. So my father died of prostate cancer. The sister of my mother died on bone cancer, and a cousin died on of lymphoma. And so there are a couple of other cases. Other people survived, but that kept me thinking, and we were at Roche already, we were defining the challenges in cancer diagnostics, and the cancer diagnostic challenges are that you have to detect the cancer earlier, of course, but there's another very important reason. That is the need to very precisely define the therapy for this particular person, for this particular patient, and in addition to follow this therapy. Are there any changes coming up, and so on? And at Roche, I was already working on a cancer diagnostics at a future technology. It's called liquid biopsy. So you take a blood sample and you analyze the blood sample. And we were analyzing a number of technologies out there, and found out that those technologies do not work good enough. And therefore, I had an idea how to improve this. This was too risky for Roche to develop a complete new diagnostic platform, of course. And so I founded and co-founded this company and set it up. And so far, it's looking promising.
What specifically does your innovation, what kind of problem does it address?
So there is first the problem to detect the cancer earlier. So this technology is able to do this. The second is that you can much more precisely characterize the tumor in order to define a good therapy, the best therapy. This is number two. And the number three is that you can monitor, because it's a blood sample. You can take a sample any time. So you can do it repetitively. So you can really follow the therapy. You can see, are there any adjustments necessary? Because if there are resistances coming up or something like this. And we do this cancer specific, I should say. Yeah. So is it breast cancer? Is it colorectal cancer? Currently, we focus on breast cancer.
So who specifically experiences the problem that you're addressing? And can you give me an example of how someone might benefit from your technology?
Yeah, so the patient and the physicians in particular can benefit from this technology quite a bit. There are several examples of what I can say about this. Is, for instance, the sister of my mother. She had, as mentioned, she had bone cancer, and for instance, this was detected very, very late, so late that she had a arm. Yeah, so she was nothing feeling here with the fingers. And if our technology should be able to detect this much earlier. And there's another example we had was with somebody who had pancreatic cancer. Pancreatic cancer is usually detected very late. And this person, a friend of mine, he had already a number of metastases around the lung and so on, was way out late. And this would have been a very good opportunity to detect this much earlier and to do something. In particular, because you cannot take a tissue sample with pancreatic cancer. And this would have been a very, very good opportunity to help such patients.
Share with me in very, very simplistic words, what does your product do?
So one should realize if the tumor is forming and if the tumor wants to grow, it connects to the bloodstream. And when it connects to the bloodstream, it leaves kind of a signature in it. That means that those are molecules then from the tumor or whole cells, so-called circulating tumor cells. Very interestingly, these cells contain all the information necessary. So it contains also information about possible drug targets. But the problem is those are very rare, those cells, and they are heterogeneous. And we have evaluated many of the current technologies about their sensitivity, how to detect those cells. And they could not detect all of those cells. And we are now finally able to detect all the cells in a blood sample. So we detect one cell out of 40 billion blood cells. Yeah, so that's very interesting and even amazing, and gives you a good picture about the sensitivity of this.
Is this a test that will be able to detect and stage the cancer or no?
It will, I think in future, it will not be necessary really to stage the cancer, because you need to get a picture of how to fight against the cancer. And those are the most important information. So we probably will also be able to detect, is there a metastasis there and where the metastasis is, and to bring this information also into therapy. That's a very important point. And keep in mind that we can follow the therapy. And the more modern therapy currently is going into the direction neoadjuvant. So you don't make surgery first. You first fight the cancer directly. And this is also very much where we think we can use our technology.
At this point, what are the biggest challenges that lie ahead?
The biggest challenges are currently technical-wise, is we have to complete the instrument. We are using an instrument, so it's that we have it needs to be faster. We want to have the assay done within 2 days. That the physician has the idea, can define the therapy, and we have to improve this instrument, and we have to bring everything into the production. That's also a major step.
So do you have any last thing that you'd like to share, or anything else that you'd like to say?
It's very important to realize that a much more sensitive and detailed cancer diagnostics is the prerequisite for the better therapy. And that is also recognized by the FDA. We know this. There's an organization in the US. It's called BLACK, and they do already a very nice job in respect to the FDA, because the FDA had very difficult to approve anything in this area. And they are now much more interested into this. Actually, they approached us one time. They approached us because they were asking from the diagnostic point of view, are there any assays which can define the tumor much better? And they asked this because of pharma industry. The pharma industry exactly needs this to define a better therapy, or better drugs. Yeah, and this is something. There's a game change. So you need a better diagnostics now to produce or to develop better therapies.
Thank you for watching the Lowdown on Cancer presented by the Cancer Fund. To learn more about what's coming in future episodes, visit us at www.cancerfund.com. While you're there, check out past episodes and be sure to click Join Us to become a member of the Cancer Fund community. As a member of the community, you'll receive early access to new interviews as well as other announcements. Make sure to connect to all of our social media channels so you don't miss a thing. In addition to getting early access to new episodes of the Lowdown on Cancer, Cancer Fund members play a critical role in driving cancer innovation forward so that one day we can more than hope. We can actually beat cancer. Thank you for joining us on this journey.